Welcome to this GCSE Biology revision video on organization and the digestive system. We will explore how the human body is organized, from the smallest cell up to the entire digestive system, and how food is broken down step by step. This video covers 4 main areas: the levels of organization, the digestive system's journey, the role of enzymes in digestion, and key exam tips. We'll start by looking at how the body is structured. Chapter 1 focuses on the levels of organization. This is about how the body builds complexity, moving from single cells to entire organ systems working together in harmony. Cells are the basic units of life. All living things are made of them, and they carry out all fundamental life processes. Key features include the cell membrane, nucleus, cytoplasm, mitochondria, and ribosomes. Tissues are groups of similar cells working together for a specific function. We have muscle tissue for movement, epithelial tissue lining surfaces, and glandular tissue that produces and secretes substances like enzymes. Organs are structures made of different tissues working together. For example, the stomach uses muscular tissue for churning, glandular tissue for acid and enzyme production, and epithelial tissue for lining. Organ systems are groups of organs performing major functions. Examples include the digestive, circulatory, respiratory, nervous, and musculoskeletal systems. The hierarchy of organization is a frequently tested sequence. It moves from cell to tissue to organ system. Be sure to learn this order and an example for each level. Chapter 2 is about the digestive system. This explores the journey food takes through the alimentary canal, how the body breaks it down, and absorbs the necessary nutrients. The digestive system breaks large, insoluble food molecules into small, soluble ones for absorption. This involves 2 types of digestion: mechanical, which is physical breakdown, and chemical, which uses enzymes to break chemical bonds. Food travels in one direction: mouth, esophagus, stomach, small intestine, and large intestine. Each region is specialized for a different stage of digestion or absorption. Digestion starts in the mouth with both mechanical and chemical processes. Mechanical digestion by teeth increases the surface area. Saliva moistens food to form a bolus, and salivary amylase begins breaking down starch into maltose. Amylase, a carbohydrate enzyme, breaks down starch into maltose. It works best at neutral pH in the mouth, but its activity stops in the acidic stomach. Pancreatic amylase continues digestion in the small intestine. The esophagus is a muscular tube for transport only. It uses rhythmic muscular contractions called peristalsis to push the bolus to the stomach. No digestion occurs here, as it lacks digestive enzymes. Chapter 3 covers the stomach, a powerhouse of chemical and mechanical digestion. Its primary roles are breaking down proteins and preparing food for the small intestine. The stomach performs mechanical churning, using its muscular walls to mix food with gastric juice. Chemically, it produces hydrochloric acid to kill bacteria and activate protease enzymes, which begin breaking down proteins. Protease enzymes, primarily pepsin in the stomach, catalyze the breakdown of proteins into amino acids. Pepsin works optimally in the stomach's highly acidic environment of pH 2, maintained by hydrochloric acid. Chapter 4 examines the liver and bile. The liver is vital for digestion as it produces bile, an essential fluid for breaking down fats. The liver's main digestive role is continuously manufacturing bile. Bile is then stored in the gallbladder and released into the small intestine. It is alkaline, which helps neutralize the acidic chyme from the stomach. Bile has 2 key functions. It neutralizes stomach acid, creating optimal conditions for intestinal enzymes, and it physically emulsifies fats, increasing the surface area for lipase enzymes to work. Remember, bile is an emulsifier, not an enzyme. The gallbladder stores bile and releases it when fatty food enters the small intestine, triggered by a hormone signal. Even without a gallbladder, bile is released directly from the liver, although fat digestion might be less efficient. Chapter 5 focuses on the small intestine. This is the main site where chemical digestion is completed and where the absorption of nutrients into the bloodstream occurs. The small intestine is about 6 to 7 meters long and has 3 sections: the duodenum, jejunum, and ileum. The duodenum completes most chemical digestion, and the jejunum and ileum are primarily for nutrient absorption. The inner walls of the small intestine are covered in villi and microvilli, which dramatically increase the surface area for absorption. Villi have rich capillaries and lacteals for rapid transport of nutrients. Glucose and amino acids are absorbed by active transport and diffusion into the capillaries. Fatty acids and glycerol enter the epithelial cells, are reassembled into fats, and absorbed into the lacteals. Water and minerals are also absorbed here. Chapter 6 covers the large intestine. This is the final stop in the digestive system, where remaining water is recovered and waste is prepared for elimination. The large intestine's main role is the absorption of water from indigestible material, compacting it into feces. It also houses beneficial gut bacteria. Feces are stored in the rectum and expelled via the anus. Chapter 7 focuses on enzymes and digestion. We'll summarize the essential enzymes that act as biological catalysts, making digestion possible. Enzymes are specific, reusable biological catalysts that speed up the breakdown of large, insoluble food molecules. They are also sensitive to changes in temperature and pH, which can cause denaturation. The three key digestive enzymes are amylase, protease, and lipase. Amylase breaks starch into maltose. Protease breaks proteins into amino acids. Lipase breaks lipids into fatty acids and glycerol. The pancreas produces all three for the small intestine. Without enzymes, the chemical breakdown of food would take far too long to sustain life. They are vital catalysts. Enzyme activity is dependent on optimum temperature, about 37 degrees Celsius, and an optimum pH which is different for each enzyme. The pancreas is a vital organ that produces pancreatic juice, containing all three major digestive enzymes, amylase, protease, and lipase. It secretes this mix into the small intestine to complete digestion. It also has a hormonal role, regulating blood sugar with insulin and glucagon. To summarize the digestive journey, mechanical and chemical digestion starts in the mouth. Food moves through the esophagus to the stomach, where proteins are broken down. The small intestine completes digestion and absorbs nutrients efficiently before the large intestine absorbs water. This final section provides key exam tips. Mastering these points will help you secure top marks in questions about organization and the digestive system. Remember the hierarchy: cells to tissues to organ systems. Crucially, know that bile is not an enzyme, and always explain the consequence, such as how villi increase efficiency of absorption. Use this checklist to ensure you can confidently state the hierarchy of organization and give examples. You must be able to name all organs and enzymes and describe their function, location, substrates, and products. For quick recall, the small intestine is 6 to 7 meters long. The stomach is highly acidic, around pH 2. The three key enzymes are amylase, protease, and lipase, and the optimum temperature for human enzymes is 37 degrees Celsius. Good luck with your exam. Remember to revise systematically, practice past paper questions under time conditions, and most importantly, apply your knowledge to explain unfamiliar scenarios.